welcome you back to Global Education Zone. And today in our biology class, we're talking about the cell and its environment. So we'll talk about diffusion, osmosis, turbidity, placidity, hemolysis, and plasmolysis. So let's dive in. Now, diffusion is basically the process whereby a substance moves from a region of higher concentration to a region of lower concentration until equilibrium is reached. So equilibrium is reached when the concentration of both regions are equal. So that's what we're defining now, diffusion. And it can be observed when, if you have a yam. When I was back in secondary school, our teacher got a yam, a giant, a big yam, and then he bore a hole in the middle of the yam, and then a salt solution was inserted into the yam, and then after some time it was noticed that that salt solution had spread inside the yam. So it has, that means that's called diffusion. Because why is that? Because the salt solution is more concentrated than the ordinary yam. So the yam cells, they are not very as concentrated as the salt solution. So that diffusion occurs because the salt solution, the molecules of the salts have diffused from that salt solution into the molecules of the yam. That's the yam and the spaces between the cells of the yam. So that's called diffusion. So basically, when substances move from region of higher concentration to lower concentration until equilibrium of concentration is achieved. Next, we'll talk about the osmosis. So osmosis is basically the process whereby water moves from a region of higher concentration, or rather, let's say water moves from a region of higher water potential to a region of lower water potential until the potential of water is equal, and it passes through a differentially selective medium, differentially or partially selective medium. So in osmosis, water is moving from a region of lower concentration of the solute to a region of higher concentration of solute. So basically, you are moving water from a region here of a weaker solution. So when you have solute particles, and this is when probably this is a concentrated medium, and then you have a differentially permeable membrane, then you have another one here, and this is partially concentrated. So your water will move. So osmosis is the movement of water, while diffusion is the movement of particles in general. So in osmosis, water will move from this highly concentrated region or of lower concentration of water to this region of higher concentration of water. So rather, basically, it will move from a region of lower concentration of solid to a region of higher concentration until the concentration is equal. So basically, in osmosis, you move from a region of lower concentration to a region of higher concentration, but in diffusion, you move from regions of higher concentration to lower concentration. So there are principal differences between diffusion and osmosis. In diffusion, you move from higher concentration to lower concentration. In osmosis, you move from lower concentration to higher concentration. Like water moves from this lower concentrated to the higher concentrated region. Next, diffusion occurs in gases and liquids, while osmosis occurs only in liquids. Now next, diffusion occurs in living and non-living things, while osmosis occurs only naturally in living things. Next, lastly, we have the fact that in diffusion, you don't need a differentially or partially selective medium, but in osmosis, we need a differentially or partially selective medium. So this will be the medium that's partially selective. Next, we'll talk about plasmolysis. Now, plasmolysis is the process whereby plant cells burst when they are placed in a hypertonic solution. So basically, those hypertonic solution is a solution that contains more of the solute. Assuming this is a plant cell now, and this is the particles of this component of the cell 1, cell whatever. Now, if this plant cell is placed in a hypertonic solution, the cell is going to be plasmolyzed because it is placed in a solution whose concentration is higher than its concentration. So water will move out of the cell, and as a result of that, the cell membrane will shrink, and then after plasmolysis, the cell will look like this. So the cell membrane shrinks, and the cell cytoplasm is lost. So that's what plasmolysis is. Then let's talk about hemolysis. Now, hemolysis is the process whereby red blood corpuscles burst when they are placed in a we have hypertonic and we have hypotonic, right? Then when they are placed in a solution of too much water, or they are placed in a solution of lower concentration. So if they are placed in a solution of lower concentration, the red blood cells will absorb water. So in plasmolysis of plants, the water actually goes out. but in hemolysis, water actually comes in. So when the water comes in, this cell wall becomes so tuggy that it reaches a point where it's actually bursts, and then the water eventually goes out. That is what hemolysis is. So this plasmolysis, water goes out, and then in hemolysis, water comes into the cell, and this is as a result of difference in concentration gradient. Now there are 5 conditions for diffusion, which I have formed the mnemonic for that. The first letter of the mnemonic is it depends on the surface area. Now diffusion occurs in liquids and gases, right? So now if the surface area is larger, then it will be faster. So that's why diffusion in gases, which have a large surface area, is often greater than diffusion in liquids. Then we have the fact that it depends on temperature. So diffusion depends on the temperature of the medium. So if the temperature is high, then diffusion will be high, but if the temperature is low, diffusion will be low. Then we have the concentration gradient. So then we have the fact that the concentration gradient is the difference in concentration between the diffusion mediums. So if the first medium is more concentrated than the second, there will be diffusion of material into the second. But if the second is more concentrated than the first, there will be diffusion into the first. So it's just like wanting to attain a balance. If you have if you have much of people in a city and they have less of people in the other city, then naturally people want to move into the less populated city because they want to reduce the pressure on the resources in the first city, right? Now that's the same thing here. So concentration gradient is just like difference in concentration. Concentration is like how much of something is in a medium. So if there's a difference in the concentration, people want to balance it out, or rather molecules in this case. Then we have the fact that diffusion depends on the nature of substance. So if the substance is a liquid, it will be fast. If it's a solid, it will be slower. Then if it's a gas, it will be fastest. So those are the properties of diffusion in different mediums. So now next we'll talk about turbidity and flaccidity. Now turbidity is the process whereby a plant cell or a living cell has absorbed water to the point whereby its cell membrane or cell wall is stressed to its maximum. So that is what how plants stay upright. So when we see a plant, just like a maize plant now, you will see a maize plant, or sorry, not like a beans plant. So the reason why this beans plant is upright is because this the cell of this plant, the cell in these leaves, the reason why these leaves can stand on their own without being supported is because the cells of these leaves are turgid, because they have absorbed water from the soil and their cell walls have become stressed to the maximum. But in a situation of drought, when there is no water, such cells begin to lose water due to sun and evaporation, and so the plant becomes something like this. So when you see cells, when you see plants like this, it's called wilting. So this is called wilting. And in wilting, the plant cells have lost so much water that the cell wall collapses. So there's no longer a supporting medium for them. So I hope with this short explanation on diffusion, osmosis, hemolysis, plasmolysis, flaccidity, and turbidity, you've been able to understand the cell and its environment. Thank you for watching. Don't forget to like, share, and subscribe, and be a light to your generation. Thank you.